So thanks everyone for joining the PhUSE Computational Science webinar. I'm very excited about today's session as the CS collaboration enters its fourth fiscal year, which is the time between CSS meetings, and I need to give credit to Sue Behaven, who's one of our representatives later, for coining that phrase. There's actually been approximately 20 deliverables produced by our working groups. And today we're very pleased to welcome two FDA panelists who will speak directly to the collaboration and its impact on the FDA. In addition, we have two working group panelists that will unveil two new deliverables that address topics in the FDA's Study Data Technical Conformance Guide. However, before we get started, I'd like to just kind of go through a few housekeeping items. So we will have Q&A at the end of the session. If you have questions, you can type them into the question functionality in the PhUSE webinar. You can feel free to type questions or add your questions at any time during the webinar. And again, like I mentioned, they will be addressed at the end. However, if you don't have time to get to all the questions, we will actually answer them offline and post the responses on the PhUSE website. So as you probably just heard, the webinar is being recorded. So if you're not able to stay for the whole session or you have colleagues that may be interested in these topics, the recording will be posted to the PhUSE website and then made available for future viewing. So I'd just like to make a few introductory remarks before we get started with the more important presentations today. So you may have heard your peers talk about something called FDA PhUSE or the PhUSE CSS. What most likely they're referring to is the PhUSE Computational Science Collaboration. So in 2011, 2012, the FDA and PhUSE did collaborate to execute a different type of meeting, or working meeting. And in March of 2012, the first PhUSE Computational Science Symposium occurred. This collaboration was designed to provide an open, non-competitive forum for regulatory agencies, both the FDA and other agencies like the EMA or PMDA, as well as industry, academia, and technology providers to work together on projects that tangibly address issues in the industry and regulatory space. And as Steve Wilson has said quite a few times, this collaboration is really akin to building the plane while flying. So I feel we've actually done a fairly solid job of building the plane, and over the past 4 years, the collaboration has produced approximately 20 deliverables, including the examples listed below. So when we talk about that plane, it's really comprised of two different entities. You have the working groups and then the annual meeting. And it's really those working groups that are doing the building of the plane. The collaboration currently features 5 working groups, each focusing on a different topic, but also collaborating across projects as the opportunity arises. You will hear from representatives from the Optimizing These Data Standards working group, as well as the non-clinical working group later in the session. Finally, all of the work that's being done on the plane is actually cataloged in the PhUSE catalog of working group deliverables. It provides the community a single point of access for all final work packages that have been developed by within the collaboration. The catalog is accessible from the PhUSE website and provides a brief description of all work packages as well as a hyperlink to the work packages itself. So with that framework in mind, I'm now going to turn the presentation over to Crystal Allard, the first of our two FDA presenters. So let me just turn the presentation over to Crystal. You should now have a presentation right now. Will she boots up her presentation? Let's give her a brief introduction. So Crystal is a special assistant to the director of the Office of Computational Science at CEDAR FDA. She's the program manager for all collaborations within the OCS and also serves as the program director for the Jumpstart service. Crystal is the FDA liaison to the PhUSE Computational Science Steering Committee, as well as the Planning Committee, and a founding member of the Emerging Technologies Working Group, and currently leads a data visualization project within the Emerging Trends in Tech Working Group. Crystal has worked at the FDA since 2008 and at the OCS since 2012. So Crystal, the floor is yours.
Thanks, Scott. Good morning, everyone, and thanks for joining us. Scott and Chris asked me if I would mind sharing a little more information about a group that I run here at FDA that I call by turns an internal FDA PhUSE users group, and sometimes I call it a PhUSE FDA participant support group, depending on the focus of that meeting. And so I think there are people in the PhUSE organization who know that this group exists and know that I run it, but I'm not sure if people really understand what they do. So I'm going to try and share that information with you today. And you know, while we're here, if everyone would look to the bottom right of my slide here, we have a new logo for our Office of Computational Science, and I think it's beautiful, so I thought I would share. So, you know, as a real sneak peek, this is the invitation that I send for our monthly meeting. So everyone gets to see exactly this, and I tried to keep it lighthearted. It's also, the actual invitation is linked. And this was just my attempt at getting people to join, because as you'll see later, we actually have a very large list of folks at FDA who have participated previously or are currently participating in a PhUSE working group or a project team. And they're all very busy, so this was my attempt at getting them. When participation starts to seem a little bit light, I actually have a tendency to track people down individually and get them to come. So we have, our mission is twofold. It's the fun stuff and the hard stuff, and I thought I would start with the fun stuff first. So our standing agenda includes PhUSE updates that we gather from blogs, newsletters, conferences, single-day events, etc. So we share the dates of upcoming events, and then we share the materials from previous events. If there's anything particularly interesting that happened at a conference, we try and share that. We try and share experiences participating in those things, and we also pay attention to, you know, what gets updated on the PhUSE news site, etc. So, you know, I know that people don't have time to read all of those things on their own, so I try to read them and share what might be pertinent to them. The other piece that we do every time we meet is announce any new PhUSE project. If there's a new project that just got started, it's not always easy for people to know when that happens, and because I sit on the steering committee, I always know, so I make sure to share those. And then we also try to see if we can get folks to participate in those. So if I know of an individual who is particularly relevant to a new project, I might approach them specifically. Otherwise, we just, you know, share the existence of the project and see if there are folks who are interested in joining it, and sort of help them understand that they're not alone should they want to, that we're willing to help them get in contact with the project organizers. You know, not everyone at FDA understands how to get onto a PhUSE project team, so we try to share that experience with them. And then we move through updates from each of our project participants. So I have a project manager that just recently had to stop working with me, but she did a really good job, and I will keep doing it, of gathering updates from each of the participants on each of the projects. So you'll see I have a whole list of everyone who participates, and I'll share that with you later. So we actually know who in FDA participates in which project, and we approach them each month and ask if they're willing to provide an update for the group on what their project is working on, and how it's going. And then we also use this group as a place to solicit participation requests. For instance, the PhUSE Computational Science Symposium that happened in March in Silver Spring, the poster judges from FDA were both found through this group. So we use them as sort of a group of folks that we know are interested in the PhUSE collaborations and who might be willing to participate in other ways. So we also bring, you know, speaking engagements if there's a webinar that someone might be, you know, a good participant in. We also try and gather folks using this group. So the other thing that Scott, you know, showed the deliverables catalog. We make sure that if there is a new deliverable available for review, that it's shared with that group. So I routinely bring up all of the resources that are available through the PhUSE website and the PhUSE wiki, including the deliverables catalog. And whenever there's a new deliverable, I not only share that it's available with the entire group, but if it's something that is pertinent, or if there's, you know, for instance, if Bob Dorsum, who works on the non-clinical team, has a deliverable that he would like someone to review, he often will bring it to the group and request that they take a look informally before it's finalized. So this is when there's a draft deliverable available for review, we try and gather informal review through that group. That includes new scripts available in the repository and any other deliverable that might come out. And then when there's a final FhUSE deliverable, we make an announcement through the group, and I'll share in a little bit what else we do with those, which is a little more involved. And then lastly, any updates from the FhUSE steering committee or planning committee are shared through this group. Again, because there are only a few of us at FDA who attend the steering committee meetings, and there are many people who participate in the project team, so we try and act as a link between the two. Okay, so this is the hard stuff. We developed a process a couple of months ago. I want to say maybe 6 months ago. Maybe it's getting on more than that. For more formally and more transparently sharing what we do with FhUSE deliverables. And we haven't done a great job of communicating that, so welcome to the first formal communication of how this process works. The good news is that we worked with the Office of Chief Counsel and the Office of Regulatory Policy at FDA and CDER, and they have vetted this process and blessed it. So it's I think it was a really good collaboration between us and them. So, and also a thank you to Helena for allowing me to use her present graphic, because the idea of a FhUSE deliverable as a little packaged present to FDA was all her idea, and it was a great one, so I've stolen it. So the FhUSE project team provides a deliverable to the FhUSE steering committee, and the steering committee reviews it and forwards it to the FDA. So that process is a little more involved, as Scott and Chris can tell you. But in general, the steering committee doesn't do a terribly in-depth review, because we think that the project teams know what they're doing. So, often it's just sort of a brief review, and then once the project team agrees that the deliverable is ready for FDA, the steering committee is the one who will let me know in particular, and I will bring it to the group. So as you can see here, there are a number of different types of deliverables, and I think you all understand that and know that pretty well. So when a FhUSE project is sent to FDA from the steering committee, the first thing that we do is bring it to the FhUSE users group internally. And at that point, we make a decision about whether or not the deliverable is something that FDA thinks they might be interested in using at some point, in part or all. And that decision gets made in the very beginning. So if the idea is this is an SDRG and we think it could be really useful, then we'll take it on. If it's not something that we think is terribly useful for us, then we can say no thanks, and we don't have to do anything else, and we'll just let the steering committee know that that's not going to be something we review. It should be noted that we haven't done a very good job of stating yes or no thanks in the past. FDA has been really a black hole, and so we're working on a website where we'll start listing the ones that will be under review, and that way you should know whether the no thanks or the yes please has happened. So the other part that happens as a deliverable comes into that group is that we identify a champion for the deliverable. If there's no one in FDA who is willing to champion the deliverable and usher it through the rest of the process, it's kind of a non-starter. And so we've seen that things that we think might be useful may sit. So I work pretty hard at identifying a specific person, and I do it through this group. If there's no one in the group, I might even spend a little time trying to find someone outside of the group or someone who might want to join our group to be the champion for the deliverable. And once we've identified the champion and we've all agreed that yes, this is something that we think could be useful, we work with ORP and OCC again to publish an FR notice, and it's a notice of intent to review. If you go to the FDA website, you can see that there are some of these as examples of other groups in CDER and FDA using recommendations from outside groups to be incorporated into guidance. And the way they do that is they issue this public notice of an intent to review. And that allows industry or anyone who wants to comment. FDA can comment on them too. And we take all of those comments, and that becomes a part of the FDA evaluation process. So I'm not going to go into what our FDA evaluation process is, just keep in mind that there is one. It's currently being piloted. It's not final, but it's happening, which is a big step, and it's very exciting. So the FDA evaluation team at this point hopefully will go through their steps to identify how they want to incorporate the guidance and not whether, but the opportunity still exists that they could ultimately decide no thanks and send it back to the steering committee. And again, the fact that there will be a feedback loop at all is a big improvement on what has existed previously, even though that would be a real shame if we decided we wanted to review it and then ultimately send it back. So then the other option is that we determine that this is something that is useful, and it could be incorporated into any number of FDA policy documents. This could be a draft guidance, it could be a standalone draft guidance, it could be incorporated in part into an existing guidance. It could, there are a number of other options, but those are the major ones. And if that happens, it'll have to go through the formal FR notice process. So if a guidance is revised, then FR notice is published. If a guidance is published, then FR notice is published. So at that point, it would follow all of the formal processes. So I think I've worked through the whole thing. This is just where I've broken it down into little bits. So here's the part where the FDA PhUSE users group will send it on to the FDA evaluation team. They're not necessarily the same people. There's another group inside of FDA that would be formed at this point that would do the evaluation. And then it would be published into some final guidance document or version of that. And we should note that as these are published and taken on by the FDA, there would be the web page that I referenced before would also be updated. So we're working on having an external FDA.gov website that lists all of the things that are under review and all of the things that have been incorporated into guidance, probably by links to other places. And so here's our, just for a recap, here's our beautiful graphic again. So there are a couple of things I'd like to mention about the deliverable review process, just of note. So there are GGP regulations, good guidance practice regulations, that permit FDA to seek information that's available in outside organizations such as the PhUSE deliverables. So I think it's awesome that we have a guidance to point to that allows us to be following this process. So should anyone ask, it is written formally in guidance that this is okay. And then there are a number of reasons why it really matters, right? So this is our attempt at being transparent and providing a process for handling PhUSE deliverables so that we get out of black box mode and into open and transparent communication with anyone who is interested in knowing how this works. It also provides a clear path for the FDA to use deliverables that come out of PhUSE as appropriate, rather than just one day, surprise industry with a link in a guidance. There's a formal path that can be followed and is repeatable and predictable so that people know what can and will happen. And it also ensures public notice and the ability to comment. So when we worked with OCC to develop this process, we noted that this will be a third option for comment, right? So, well, I guess this will be the second, but there will be three, possibilities for commenting on any of these deliverables that get incorporated into FDA policy. So the first is the PhUSE collaborations are available to anyone who wants to participate. So anyone who has a voice or wants to have a voice can participate there. Then we'll publish the notice of intent to review, and anyone is allowed to comment on that. And then when it comes into a guidance, that will also be published and people can comment on that. So there will be ample opportunity for folks outside of FDA to comment on the thing before it actually becomes, formal set in stone guidance. So this is our cast of characters. It's pretty long. I've first listed all the people who are sort of active members, and it's in alphabetical order so that no one feels any more or less special than anyone else. And you'll see that we actually have a large number. This is our entire list of folks, and we sort of add to it and detract from it depending on how it's happening. This is my shameless plug for all of the resources that are available from the steering committee. I share it routinely internally here at FDA, whether you believe it or not. The steering committee works really hard at creating these processes, and it's important that people know they exist and they're out there for your help, not to badger you into a formal process, but just to help you should you need it. And we have questions. One last thing before I go back to the questions. I just wanted folks to know that SDA has a Flickr stream, and we're on it. Thank you to the lovely Stephen Bamford who took these pictures for us. Questions? Scott, is now a good time for questions?
Well, we will take questions at the end. So
okay.
if you have some questions for Crystal, please feel free to type them into the GoToWebinar question functionality. And like I said, we'll get to those at the end of the presentation. Crystal,
okay.
That's excellent. That really has provided some excellent insight into how FDA is taking what's being done in the collaboration, pushing it through, and eventually some of it might end up in good. So I really appreciated you taking the time today and coming to present at the webinar. I am now going to hand the presentation, or the presentation has been handed over to Gino Pizarro. I hope you pronounced your last name right, Gino. Gino is going to talk a little bit about the SDRG, which was something that was in the experiences within the FDA, and it's something that was produced within the collaboration. So Gino is a senior consultant in IBM's Advanced Analytics and Watson Group, and has been an analyst at FDA's Jumpstart service for 2 years. During that time, his team has helped medical officers review roughly 50 new drug and biologic applications by assessing the quality of submissions and helping to kick off safety reviews. So Gino, you now have the floor.
All right, thanks a lot, Scott. And let me know if you have any problems with my audio.
Everything sounds great.
Awesome. All right, so as Scott mentioned, I'm an analyst with the Jumpstart program at the FDA, and in the last 2 years, we've helped kick off the review for medical officers for about 50 applications. And as a result, we've seen a lot of submissions, and we've also seen a lot of study data reviewers guides. So what I want to sort of leave everyone with today is that the study data reviewers guide is a tremendous opportunity to speak directly and clearly to a review team about a data submission. So I'm going to go through a few sections of the SDRG that we see this opportunity both being missed and also being taken advantage of really well. So just a quick disclaimer that my views and opinions are mine and don't necessarily represent the FDA's guidance. So my colleague Austin and I presented about the Jumpstart service at the Computational Science Symposium, but in case anyone wasn't there or didn't see that presentation, I'm going to give just a quick overview of what Jumpstart is and how specifically the Study Data Reviewers Guide fits in. So we start very early in a review process. In fact, before the data package is received by the FDA. And we have an orientation session with reviewers. We walk them through the tools and processes that we were going to do during the Jumpstart process. But once the data package is received, we spend about 2 weeks doing an audit of the data quality, which culminates in a data fitness session. And what we're doing here is we're checking across and within data sets for consistency, accuracy. We're seeing how everything's conforming to data standards, and we assess the relevance of any deficiencies and show reviewers how any of these problems might affect the review that they're trying to do. And then 2 weeks later, we do an exploratory analysis session where we run some safety tools and some tools around laboratory results and vital signs, and then of course follow up with reviewers, make sure they have everything they need. If they're going to submit any information requests, we help them put those together, and then we also make sure they have all the training resources and stuff like that. But within the data fitness session, we take a look at a lot of different things, such as missing data. We look and see if there's any duplicates that will cause problems, inconsistencies with standard units, and so forth. But you'll see the very first thing we do is take a look at Study Data Reviewers Guide in the define file, because we want to get a sense of how the metadata are, and are these documents going to be good for the review team to get a sense of what's available to them, and to get a quick look at how the data package was put together. So with the study data reviewer's guide in particular, we assess the completeness of the document, making sure all the sections were taken care of. We look at the versions of standards to look for any inconsistencies. We take a look at the SCTM and supplemental description sections to see if they're going to be useful. And then also we spend a lot of time looking at the conformance section to see what version of OpenCDISC was used and are the explanations relevant or useful. And really our assessment is all about taking a look ourselves and telling the review team whether they can rely on this document and whether it's going to be useful for them. And through our experience, as I mentioned, we've looked at about 50 applications now. Our big takeaways are that metadata, the putting together of quality metadata, it seems to be commonly overlooked. Even some of the best submissions can have some deficiencies with metadata. And metadata and poor metadata is going to equal wasted time during review. Because data without clear definition, those submissions can be confusing because reviewers really feel like they've been given a mystery box if they have a data submission in front of them without a clear roadmap to look through it or use it. And this is because explanations really are needed for an applicant's particular interpretation of STTM. So a reviewer may be looking at a submission that the interpretation of STTM is quite different from the one that they got before. And this could be because of any sponsor-defined controlled terminology, any derivations that are unique to this submission, and then of course custom data, which is likely to be in a format that a reviewer hasn't seen before. So next I'm going to go through, I picked a few sections where we think that are particularly useful for us as data people and also where we've seen the most issues. So up first, the study data standards and dictionary inventory. And this is a really simple table and section that tells you about all of the relevant data standards. And it's great to have them all in one place because a lot of the review tools at the FDA require an input of certain standard dictionaries. So you might be required to put in what version of Medra you're using, what version of STTM was used to run some standard analyses. So it's nice to have that in one place. Now, of course, the problems we've seen are versions that are inconsistent between various documents. Say the study report shows one thing, Define shows another, and the study data reviewer's guide shows another. Or versions are wrong. So say the documents are saying Medra version 13 was used, but when we validate, we see that it's really 14 or 15. And this will cause a problem having some adverse events drop out of analyses if they aren't able to be matched up. So moving on, I'm going to skip over the protocol section and go to the dataset description section. And starting with the first part, the overview, this is a question answer section that gives some really good information about the source of the data, so we can tell whether a trial is ongoing. We can see whether this data was collected originally in STTM or was legacy converted data, whether analysis data sets were created from STTM, which can be really helpful as we try to trace data back, and also whether screen failures are included. So in the SDRG completion guide, which I'll show a link to that later, there are examples of a lot of different additional content that can go in this section, and I wanted to highlight a few that we've actually seen in submissions that were very useful, we thought. So one was a really detailed breakdown of the source of data, telling you what's coming from the ECRF, what's coming directly from, say, adjudicators, what's coming from external labs or central labs. How can we tell? How's that denoted? Some information about the breakdown of where death information is located in a submission. So this is something we do in Jumpstart for every submission. We reconcile death information, but it's really nice to see an applicant put that together and showing you where they have all of the death information represented. It makes it very easy for us to validate, and it's also great for applications that maybe don't have a Jumpstart service being performed. Discussion of key efficacy data. If any domains are set up particularly for the efficacy, and how to analyze that data, how it's put together. It's really nice to see a discussion about that. And then also a discussion of reference dates, which as we are moving towards the newer versions of SDTM, this is becoming, I think, particularly important, because we do have multiple versions of reference dates now, with participation dates and exposure dates and so forth. So it's nice to see in plain language what do each set of dates mean. Maybe how is treatment emergent defined, what dates are being used for that, and which reference dates are used for efficacy analysis. Is it participation dates? Is it the regular reference start date? And so forth. So it's nice to see additional content like that. So moving on to the SCTM domains table and the corresponding descriptions. This is just a really, and once again, a lot of things about the SDRG are just simple, and that's what's great about them. It's a nice place to see all the data that's available in a submission. It's great to see where the safety and efficacy data are located. And down below the descriptions section, they list the supplemental qualifiers and what they mean, which can be really nice to see what data are available in supplemental domains, and whether a reviewer is going to take advantage of that, or whether that's going to be particularly useful to them. It's also nice when there's a discussion about how a domain is linked to others, which can usually result in explaining how the sponsor identifiers work together, or link IDs or group IDs and stuff like that. And this is a great place to put information that might be unique. So if a data set is a unique data set or a custom data set, giving a good description about what's in that data set, what's going on there. And then lastly, if a variable uses sponsor-defined control terminology, a discussion about what the values mean, what the potential values mean, and how they should be interpreted. This is a good place to put that as well. So the common issues here, very simply, if the efficacy, safety, and RELREC columns are missing, that's a shame. We've also had problems where the RELREC information is incorrect or inconsistent. So for example, if you have adverse events and it says it's related with conmeds, but then as you go down, you see that, well, clinical events and hospitalizations are also saying that they're linked with adverse events, so it's a little confusing when that's the case. And then also, it's definitely a missed opportunity when the whole description section's missing, or the section detailing the supplemental qualifiers is missing. That's a missed opportunity there. And then moving on to the data conformance summary section, this is probably where we see the most issues, the most missed opportunities. So I have an example here of kind of what we see a lot. And this is just a message saying that the adverse event start date is after the latest disposition date. And the explanation here is just as collected. And a lot of times we'll see stuff like as received from vendor or lab. No care was taken to give an explanation. Really, these are just acknowledgments. And then also another issue we see, which will be above the table, is not clearly defining which version of OpenCDIS that was used. And this is really nice because it lets us know what rules are run, what rules were not run. And sometimes we see applicants using really outdated versions of OpenCDIS, which is a shame because it misses some of the newer FDA business rules, and it also misses some of the things that were changed in the tool to try to get rid of false positives. So a good example then would be, this is the same issue. Subject 123 completed the study on January 1st, but died on July 1st, and as a result of the protocol, subjects were followed after the study for a live dead status at various points. So this one is explaining the data to someone who's unfamiliar with the nuances. It's written for someone new to the study, and the issue here, it's broken down based on at a subject level. So we really like to see if a conformance finding is is different based on groups of subjects or different lab tests or vital tests, it's nice to have that level of granularity, splitting the explanation up by those groups. And I like to say, if explanations are clear and comprehensive, it's really going to diminish the need for information requests, because everything we need to know is right there, and that really helps the review teams, and it really helps us understand what decisions were made with the data. So I just want to leave with this, that you should definitely take advantage of the resources on the PhUSE wiki for all projects, but the SDRG package is a great package, and it includes templates that you can use. As I mentioned, the completion guide, which is very thorough and easy to follow, and then some samples. And there's a link, but you can also just search PhUSE wiki study data reviewers guide, and you should be able to find it very easily. So I'm going to pass it back to Scott.
Excellent. Thanks for that presentation, Gino. I think that really helped to provide some insight into how the FDA is actually using SDRG in context they are finding helpful. So let me turn the presentation over to Jane. So now we're going to have presentations from two working groups that are about to unveil some deliverables that directly address some items in the study data technical conformance guide. And the first is Jane Lozano to speak about the study data standardization plan. So Jane is an integrated database consultant at Eli Lilly. She has been with Lilly for 21 years, starting in their oncology department, supporting multiple compounds, and has been involved in multiple submissions. Jane attended her first PhUSE conference in 2014, and she's jumped right in. She is co-leading the study data standardization plan project, as well as the legacy data conversion template project. So Jane, I'm going to turn the presentation over to you.
I had to unmute myself. Thank you, Scott, and welcome everyone to the webinar. As Scott stated, I will be giving you some information on what is going on in our working group that is working on creating a template for the study data standardization plan. Let's see if I can go into, there we go, into full screen mode. What I want to cover is the background of the plan and who our team members are, what we're doing within the team, or the makeup of the group, the state of the plan, where we are now, and then what the timeline is for publishing or getting the plan out ready for review. Background, let me see if I can, I don't know if I can move this or not. Oh, not. I don't want it to hide what I'm showing. Sorry, I apologize. I hit the wrong button. The SDSP, we call it that. Everything has to have an acronym, so we are calling this the SDSP working group. We was actually formed at the 2014 PhUSE conference. It is described in section 2 of the technical conformance guide, and you can read what it states. They wanted someone to help lead this group, and this was my first PhUSE conference, and I said, sure, why not? I wanted to get involved, and I have been involved ever since. And now I can't page down. Here we go. The deliverables that we are going to have out of this working group is a template, completion guidelines, an example document, and also we are going to produce a document for regulatory communications. And I will let you know what that means in a couple slides. Our team members are made up, we have 38 team members, made up from multiple pharmas and consulting firms, FDA representation, we have clinical, non-clinical. We do meet every other week. We actually met this morning right before this meeting. We all meet to collaborate on the template, but then we also have subgroups. Because of this number of people, it's a great way to utilize everyone and have different subgroups working on other deliverables. The subgroups that we have are the completion guidelines, and Elizabeth is going to lead that once we get a good enough template, a baked one as I would like to call it, to start on this. We don't want to have to redo things by starting on completion guidelines and then having to go back and redo it. An example document, Lisa is leading that, and the FDA communications subgroup, and that is led by Anne. And the purpose of the FDA communications subgroup is to help answer the who, what, when, how types of questions. When do you create this? Who creates it? Who's responsible for it? How do you communicate? That type of thing, the process behind creating this plan. This plan could be done at pre-IND. Sometimes it's done at the end of phase 2. It depends upon where the development of the compound is in. But the purpose of this document is to help guide sponsors in knowing when to create it and just the whole process involved. We felt that that was very important, not just giving a template, like you know when the SDRG is supposed to be created, but this plan is a living document throughout the life of the compound, and we want to help provide that guidance as far as when to create it and who creates it. The state of the plan right now is that we discussed the template. We actually had a template. We've been working on it since 2014. When we met in March of this year, we discussed the template. We discussed that this is one plan per compound per indication. Therefore, studies could be part of multiple plans. And we realized that, and that is what we have said this plan would be. However, one plan per compound per indication. Then in May of this year, FDA issued a recommendations document, which was wonderful to get that feedback, that input from FDA on what they're looking for within this type of a plan. The template that the SDSP team had was updated per the recommendations document. We have some extra things in there that we think is important that should be in there when we have those discussions about standards. But we did incorporate what was in the FDA recommendations document. We are finding that definitions are very, very important and must be in alignment with FDA. And I will show you an example. I will show you the template, and we were actually working on definitions this morning as to define what is a study start, what's a completed study. And that is something that we will have in our template that cannot be changed, and that we have to make sure that we are in alignment and everyone is in alignment with what the definitions are. We're also going to put in the plan, and we have, if you can call it legacy data, non-standardized data, and data that does not conform to the current standards, we have a place in the plan to indicate. Sometimes we're going to live in a legacy world for quite a while until everyone starts submitting data in a standardized format. And we have to be able to have a way to have that conversation with FDA on if we have legacy data, and even if it's in a version that's not supported, SDTM 311, it may not be supported by the FDA anymore. But to have those conversations as to this is what we want to do with it, and then document also within the plan these conversations that had taken place, and have the agreement written in there so that we can go back to look at it later if we need to for reference and see what was agreed upon. Our timeline, and it's somewhat aggressive, but since we've been working on it for quite a while now, I would like to have a template ready for the next level of review by the Optimizing the Use of Data Standards leadership team by the end of June. And then once we do that and get that review done, then shortly thereafter we can actually start on the completion guidelines and the example document, which is going to be very important. I know we have one for the SDRG, the ADRG. These are, I think, very important documents to have. I would love to have a first draft available for review outside of our group by September 1st. Anne is working on the FDA communications document, and she's doing a wonderful job, as everyone else on the team. They're doing a fantastic job to have that document ready also we're hoping by the end of June. I would love to have deliverables completed by the end of the year. I do admit I am new to the review process, and I don't know if that's possible, but I'm shooting to get this done, especially before the next PhUSE conference. I would love to have this done, but I will need help in knowing the proper route. And Crystal, thank you for showing your slides. Those were wonderful, and what needs to be done. But it's what I hope to accomplish. There's our list of team members, and you can see we have a lot. And I love having all the team members. They're representing all the different groups, and FDA representation is so important. And I appreciate everyone that is a part of this group. I know some of them are on this webinar right now, and your input and just collaboration that we've had has been really fantastic. And after that, I will show you briefly, because I know we don't have a lot of time and I know you want time for questions, is the main crux of our study data standardization plan is to separate it out between clinical and non-clinical. And I do apologize, I don't know how to move this over. Here we go. You get it out of the way so I can show you, so you can see the whole plan. But it is pulling in what the FDA recommendations document has as far as a study identifier, a title, a type. We added the status. We changed it a little bit, where the FDA recommendation document had a section for planned studies and completed studies. We are putting it all into one table and to have a study status within the table. We also think study start date is important, because as you look at the data standards catalog, it's very important to know when standards are going to be retired, and that you can plan that, and we have these conversations for what standards you're going to use. We also have a column for legacy data, and I'll tell you, we are coming up with that definition of legacy data, and we'll need to work with FDA on this too, of how do you define legacy data, but at least have that conversation, whether you have it or not, whether you're going to convert to standardized data, you're not going to convert, and you can have that conversation. For non-clinical, their exchange standards are going to be different than the clinical world, and the information that is being gathered is pretty much the same, except for study type versus study design. And the table is split out by phase 1 studies, phase 2, phase 3, phase 2, 3, you can have phase 1, 2, and the ability to put in those combinations that are there for the sponsor. The exchange standards, we originally had columns for each of these, but based on the FDA recommendations document, they put it all into one, which is very compact, and it looks good like this, to put in your SDTM version, your IG, and define. Do the same thing with ADAM, and then all the different terminology standards that are in the data standards catalog. There is also a section to explain why you're not going to conform to standards, which is in the technical conformance guide, saying that the plan should have a section, you should be able to describe why you're not going to conform to standards. And then we also have a section for the regulatory discussions, and that's where Ann's group, the FDA communications group, is writing the document for the process, and then we can then document those discussions that happened and what the agreement is. That is it, because I know you want to be able to have time for questions.
Well, thank you.
So Scott, I will send this back to you.
Great. So it looks like this group has got a lot of work done, and we've made a lot of progress. It'll be great to see when that finally comes out as a draft for folks to review and get some feedback on that. So I am now.
Yes,
sorry.
Oh no, I said yes. It's very exciting. I'm excited about having something done that we can send out for review.
So now let me just make Sue DeHaven the presenter. She will be our last presenter for the day, and we have quite a few questions. So Sue, again, is going to present on something that directly has come from the FDA's Study Data Technical Conformance Guide, a non-clinical SDRG. Sue is the Global Director of Applications and Information Management at Sanofi, based in Bridgewater, New Jersey. She's started collaboration since its inception and is the co-leader of the non-clinical working group. Sue, I will give you the floor.
Okay, thank you, Scott. Can everyone see my presentation?
Yep, that looks good.
Yep, okay, great. So I'm going to follow along and talk specifically about the non-clinical study data reviewers guide and what the working group has produced and what the status of that document is. So I'll jump right in. We also refer to the study data technical conformance guide, and I think as you've heard throughout this webinar, we refer to that guide quite a lot, which FDA published along with the e-study data guidance. And it speaks to the value of the information provided in that guide. So that's really a wonderful resource, and I recommend for people to have that handy, have the link to it handy when they have a question about how to prepare data sets. In that guide, there is a statement: the preparation of a study data reviewer's guide is recommended as an integral part of study data submission, and I think Gino described that really well for the clinical situation, and we feel that's the same for non-clinical. So a few years ago, we formed a team to evaluate whether the clinical SDRG could be adapted for the non-clinical purpose. The Optimizing the Use of Data Standards Working Group had already published their first version of their SDRG template, and that is the legacy version of what Gino showed today. And we took that example and tried to fit in a non-clinical study into that. And we published a poster on that at the CSS in 2014, giving our analysis results and our recommendations for how a non-clinical template should look. We then followed up, by then the technical conformance guides drafts were coming out. We did analysis of that, and we continue to do that to make sure that all of the guidances that have been published since then are included in the templates. And we also solicited input from FDA benefactors that we have that are partial to non-clinical review information to help us understand what we could include in the template and the guidance document in order to provide the most value. We then, at the 2015 Computational Sciences Symposium, put up our poster on what our current template looks like for feedback from poster reviewers there. And now we have produced an SDRG template user guide, an example package very similar to the clinical package. And that is currently posted as a draft on our wiki site. So the SDRG table of contents that we use for non-clinical is essentially the 6 sections recommended in the technical conformance guide to be included but not limited. So we followed that directly, and I'm going to talk to you specifically, give you some examples of the content from our guidance for sections 1, 2, 4, and 5, which are sections we've recently worked on where we had some direct feedback from FDA on what would be helpful to them for sponsors to include or study conductors to include. So section 1, introduction, is kind of basic, and it starts you off running with the study identification information. And this essentially links the document to the data sets. Which study are we talking about? And in there's an opportunity to describe to the reviewer the send data set creation process that was used to create the data sets for that particular study. Remember, we're on a study level, not a submission level. And I think this helps to provide this context that Gino had mentioned about, how were these things created, which labs did they come from, things like that. It also gives us an opportunity to indicate to the reviewer a confirmation that the SEND data sets accurately represent the study report. If needed, it can guide the reviewer to particular sections of the SDRG where any differences between the SEND data set and the final report could have a further description, because this is something that could really generate a request for more information about why something is different. In the section 2, study design, this is an area where we had a lot of discussion in the team about repeating what's in the protocol, repeating what's in the data set, and what we realized is that many sponsors that are conducting, or many labs conducting non-clinical studies, we commonly have what we call a dose group chart. Different companies have different versions of that or different formats, but they're very useful in helping to organize someone's mind around which dose groups in our mind, in our vocabulary, our sponsor-defined group code, which dose groups have which arms and which sets and things like that. So it really helps the reviewer bridge the study design description that's in the report or the protocol with how that is structured in the SEND trial design data sets. So it can look a little bit redundant to information that's in other parts of the submission, but really the overall value of it became apparent that if this is here, the visualization of the data sets becomes clearer for the reviewer. This particular diagram that we use, this chart that we used in one of the examples, is just one idea. There's no requirement for a particular format. And sometimes, as you see in study plans, this description can be textual. So this is really optional. Section 4, I think Gina went into this much more excellently than I did even here. There are, for sure, questions that are helpful, and some questions are more relevant to clinical and some more relevant to non-clinical. So in our examples and our guidance document, we tried to include particularly questions that were focused on the non-clinical situations. Here, our assumption is that probably in the derivation of the study data reviewer guides, different groups of people would be generating the clinical versus the non-clinical documents. So we felt it was good to have a clinical specific, a non-clinical specific template. And section 5, this is an area around a section to explain these conformance issues or places where the validation rules are broken. And again, I'll refer back to Gino. Thank you so much for providing such an excellent description of why that's useful. In the discussion that we had with our non-clinical reviewers at FDA helping us out in describing our guidance document, we got a bit of information that was specifically helpful for non-clinical. FDA has provided a list of validation rules for SEND data sets that's available on the Study Data Standards Resources website at FDA, and their specific rules were really helpful to us, this list, because this is structured to check the conformance of the data sets for what the non-clinical reviewers are going to do with them. And there is an open CDISC validator, there are internal validators for non-clinical data sets, and the important thing for the SDRG was determined to be that we focus on the FDA rule conformance, and there is an opportunity in there to describe which version of those rules that you're using. So you focus on those, describe the validation method or version that was used, annotate the warnings, and also optionally describe any notices. So this is an example out of one of our example SDRGs on the website. The status today. So I think now, based on the previous presentations, you'll very well understand this status much more clearly. The study data reviewers package was submitted to the Computational Science Working Group Steering Committee for deliverable review. So we provided them with all of the content that's in the package that you can see today on the website, on the wiki site. The steering committee is facilitating the internal review by FDA, PhUSE participants. So that's exactly what Crystal was talking about, and Crystal is very helpfully doing that for us. And today we're hoping that this package will be passed on for a formal public review soon. So we're anticipating that outcome of the PhUSE participant working group or PhUSE participant team that Crystal described, and hoping for the best that we'll pass on to a more formal review. So just some takeaway things to keep in mind. Really over and over, FDA reviewers that we asked for input on the non-clinical SDRG, you know, what do you really need out of this? And the advice was to focus on what's important on a study-by-study basis. So even though we're providing a template and a guidance and some examples, within the examples we're trying to show that study by study there may be different things that are important to point out so that the reviewer who's reading that document can have as clear an understanding as possible of what those data sets are that he's going to review, or she. The future, so when we roll out deliverables like this from a project team, you know, they can become obsolete if they're not maintained. Technical conformance guide is updated, which may drive some changes to these things. Various things can happen in the regulatory environment. Experience can teach us that there's some value to making an addition or a change in these templates. So the SDRG working group has agreed among us until a future date, we have no idea. We will continue to meet less frequently once it's published and keep the responsibility to update the SDRG template based on feedback we receive from anyone who's checking it on the PhUSE website and any updated guidelines from the agency. So I want to thank, we have a lot of people that participated in the development of our SDRG package. So we have a list of people from vendors and pharma companies and CROs and a lot of great contributors. And a huge thank you to PhUSE and FDA. They've been so supportive of our project. So thank you to them. And Scott, I will pass it back to you.
Thank you so much, Sue.
Sure.
Again, another example of a PhUSE project trying to make a difference in response to unmet needs. So I know that we have a lot of questions, and I just want to take time to, again, thank the presenters for coming today and for giving us a great series of presentations. And I also want to thank the PhUSE office, who we've been doing these webinars now for maybe 6 to 8 months, and they have worked tirelessly and unheraldedly behind the scenes, helping to get organized and put things together. And Tori and Tracy, thank you so much for doing this. So let me just move to the questions. So the first one will be Crystal. It's great to know the processes at the FDA, the person asked. They said they saw that there are some FDA scripts in the PhUSE script repository, and we're curious about the timelines for releasing more FDA scripts and adding those to the repository.
Sure. So we are actually willing to send more. We've been sending them in small groups. The only reason that we didn't send more in the last few months was because we knew that the script repository group was working on migrating to GitHub. and they were actually hoping to get that nailed down and finalized before we sent them any new scripts. But we talk to them pretty routinely, and they have members of their group on the steering committee that we always catch up with. And so we're actually willing to send more, whenever they're ready to receive them at this point.
Okay. I think this question is either for you or for Gino. Do all submitted studies go through Jumpstart? If not, what selection criteria is used?
Sorry, is that for me?
I could be for Gino or for you, Crystal. Do all studies go through Jumpstart?
I can start, and then Crystal, you can fill in. They do not. Right now, I think 30 applications are going through this fiscal year, and we have some selection criteria about the types of studies. Phase kind of studies we do. An analysis session kind of requires a fairly standard type of study. I don't know if you want to fill in anything, Crystal.
Hi. So I tried to mute my phone and I hit the hang up button instead, so I called back in. Yeah, no, there's no right now formal process for giving it to everyone at FDA. We don't have the resources. And as Tim mentions a lot when asked this question, the process doesn't scale. It's not like you can just get 50 more IBM analysts and then give a Jumpstart service to everyone. It doesn't really work that way. And it doesn't scale in the reverse either. You can't just start doing it more automatedly and then hand it off because of the way that the Jumpstart service is a combination of both manual and automated processes that require learning and expertise in a lot of ways. So there's no current plan to roll out the Jumpstart process as it currently exists to everyone at FDA. And we therefore have a process now by which people request a service if they want it, and we look at it and make sure, A, their data is an SDTM, because it's not always. You'd be surprised how often we get requests for the service for data that's actually not an SDTM. And then whether or not, their studies are sort of appropriate for our service. Most of them are, because our service can be modified to work on most types of studies. And then, it's a resource game. So sometimes we're very busy and we have to either put people off or say no, which is both good and bad.
Okay, so I think this next question is for both of you as well. So there's certain, we have a couple of questions with this kind of phrasing. So certain elements in the SDRG seem to duplicate information in defined XML. Is this duplication necessary, and is it useful for the reviewers?
So I'll start with that one. Yes, the duplication is necessary, and yes, it's likely useful for the reviewer. So the information that's in a defined.xml is very useful to our Jumpstart service and our Jumpstart analysts, but it's not in the most human-readable, human-friendly format, and the SDRG is. And the other piece about duplication is that, just to give a plug for the SDRG to be used in its entirety as it was intended, that team had FDA folks on it, and they worked really hard at ensuring that the information that was on it was appropriate and necessary. And so, in general, we like to recommend that folks use it in its entirety as is possible. I understand not everything always applies to every study, but where possible, it's better to err on the side of putting more in there than less, because reviewers find it very useful, and they do look, and we look.
Okay. So this question is for Sue. And what parts of the SDRG for non-clinical are significantly different than the clinical SDRG?
I probably wouldn't use the word significant for any of the sections, I would say. What I would refer to is in the 2014 Computational Sciences Symposium, we did exactly that comparison, and we published a poster there to say, you know, which parts were different. I would say in the description of the data sets, the team spent the most time in refining questions that were that would prompt the most useful information for non-clinical reviewers versus clinical reviewers. But it looks quite similar. There's a few vocabulary differences. We don't need to talk about efficacy. The non-clinical SDRG is applied to non-clinical safety studies, so I'd say maybe that could be considered a significant difference. But I think they're quite aligned, and we plan to keep them that way. In fact, with the Optimizing Data Standards Working Group, who's maintaining the clinical document, our goal is to maintain those two packages in parallel so that they're consistent and supportive of each other.
Okay. So sort of as a follow-on to that for Gino or for Crystal, has there been any experience at the agency receiving SDRGs for non-clinical studies?
So I saw that question come up, and I tried to get in touch with John Ho and Tim Kropp, but they are not answering the message I sent to them, so I do not know the answer. I'm sorry. I haven't heard of us receiving any of them. I know that, you know, as we talked about there, it's a really new document from PhUSE. We do have folks at FDA who were involved in the development of the non-clinical SDRG with the PhUSE group, but I don't know if we've seen any actually arrive here as part of a submission.
Okay. And is the FDA planning to ask for any content that's currently in the SDRG to be incorporated into CDISC standards, perhaps?
I don't think we have any plans to do that. It's not really in our wheelhouse exactly. If there are recommendations from an SDRG for CDISC or any SDO for that matter, they would come from the PhUSE project team. They have a link to CDISC and they make recommendations to them. So I don't know that we would do that.
Okay. So here's a question for Gino. Is it possible to provide findings useful or bad examples for improvement, specifically with relation to trial design modeling? So examples with that people have submitted the SDRG.
So I saw this question, and I would like to dive into this a little bit more and get some good examples, because off the top of my head, the only so I think that,
yes, I know your phone is for example, but maybe that's something we could follow up on after the webinar, and maybe perhaps get some additional examples that we could post with this session. So I think this question might be for Jane. So the timing of the SDSP is about the same as the timing of supplying the annual IND update. Would there be value in merging the two documents together? I don't know if that has come up at all during the SDSP project, Jane?
No, and can you repeat that again, please?
Sure. So the question is, the timing of providing the SDSP is about the same as supplying the annual IND update listing of planned studies. Would there be value in merging the two documents or two requirements into one document?
I would have to see what the other document has in it. The purpose of the SDSP can be done at various times. Like I said, it is a living document, but it would be worth it pursuing. I'm not sure what exactly is in the other document. Is there a way to see that?
We could follow up on that. Maybe take that one offline.
Okay. Does that make sense?
Yes, that does make sense. That makes sense to me.
Okay. Thank you.
So we just have a couple more questions. So the SDRG and SDSP are great. We're wondering if the repetition of information that's manually entered may cause discrepancies, especially for ongoing studies. If there's any thought from either the SDSP team or SDRG team about how to kind of minimize that potential risk.
Oh, this is Jane. I think there could be probably some overlap, but I thought when you actually did an SDRG, the study was completed or well, it may not be completed. It could be ongoing, and that you're submitting the data. But the purpose of the SDSP is to have discussions as early as possible, way before you create an SDRG. And then it would be, I agree, it would be important to make sure that what's in the SDSP matches what's in the SDRG based upon what was agreed upon with FDA. And we struggled with that when we were creating the SDSP of, well, this is somewhere else. Well, this information, you could find it somewhere else. And then we got the FDA recommendations document, and yes, some of that is overlap, but the whole idea is to do this early on. Sometimes that's not going to happen, but it is. My thoughts are is you create your SDSP and have these discussions before an SDRG is actually created.
Yeah, maybe let me just add a little bit to that from the non-clinical perspective. So I think the study data standardization plan, at the point that it's starting to be published and used in pre-IND meeting or something like that, we could have, at least in my organization, have started generating SDRG documents for non-clinical studies in anticipation of a future submission. And it's true, and I agree. It's maybe it's a risk. And Gino noted sometimes they see some inconsistencies where versions of the standards are noted differently in different documents on the same submission. And I agree that in the place where we say on a particular study, this is the versions of the different standards resources that we used for this study, that then has to be transcribed forward into the study data standardization plan. I like that mode better because if at the time of the generation of the SDRG, the study is done and we know what standard we're using. Where if we project on a study data standardization plan what version of standard resources will be used on a future study, that would need to be checked, retroactively verified following the conversion of the data. So there's some work there to maintain consistency until we come up with anybody have some creative ideas on how to use some sort of master system to propagate that information across multiple deliverables. We're always looking for things like that.
That sounds like another PhUSE project, I think.
Doesn't it, though?
It does. So we have one final question, and then we'll wrap up for the day. So I think this is directed to Crystal and Gino. So if a sponsor lists SBTM IG 312 as its exchange standard that has some of their data using a higher version of SBTM, such as SBTM 6.2, the draft 3.3, or even the therapeutic area user guides, where and how should this be documented? This is the first part. And the second part, is this an issue with FDA reviewers? I don't know, Crystal or Gino, if you can speak to that.
This is one of those times where I wish you could see my face. It looks very confused. So that should all be documented in the SDRG. But some of those standards are not yet listed on in our standards catalog as accepted by the FDA. So let's keep that in mind. And the information, that sort of information is very useful when going through a Jumpstart service, and also very useful when setting up an analysis, or after having tried an analysis and discovering that some things aren't working the way that you want to, and you have to try and figure out why. That's when people really go to that's when a reviewer is really going to go to the SDRG. Outside of that, we don't have much experience with that example exactly. And the appropriateness of it, I think, is probably better answered by someone like Ron Fitzmartin, because that's the wheelhouse of our friends, our sister group in OSP. Does that answer the question? I can't find it or read it, so I might feel like I may have left something out.
Sure. I think that kind of answers the question. I guess has the FDA actually seen any submissions that have used like SDTM 3.2 or the therapeutic area user guide?
I'm not entirely sure. Doug Warfield would know that.
Okay, well, maybe we'll follow up.
We haven't seen any. Gino, we haven't seen any in the Jumpstart service, right?
We have in 3.2.
Right.
I would just echo what Crystal said. If you want to put this in the study day reviewers guide, that'd be great. And more information, more granular information is probably better than less. So.
Excellent.
Careful what you wish for.
That's true.
Okay, so with that said, we'll wrap up for today. Again, I want to thank the panelists. I think this has been a great session, and thank the PhUSE office for their support as well. So we'll probably do this again in a couple of months. So until then, thank you for your time on the phone, and we'll talk to you again soon.
Thank you, Scott.
Thank you.
Thanks, everyone.
Bye-bye.